Hello and welcome to another edition of Zog Science. Today we're going to be talking about the process that forms proteins, and that's going to involve transcription, translation. We're also going to talk a little bit about mutations. So what is the function of DNA? We talked about this before. The DNA molecule contains all of your hereditary information in the form of genes. Okay, so we've got a sequence of A's, T's, G's, and C's. And different sections of that code for particular things, and those we call those genes, the particular section, whatever that is. So it's just a portion of the DNA that codes for production of a specific protein. For example, the proteins in your muscles, the proteins in your hair, whatever it is, it's just the particular protein. We call that a gene. So here's your chromosome. You've got long strands of DNA, and you've got various genes kind of in sections. They're not necessarily all right next to one another. In fact, some of them can be spaced out very far apart. But remember, DNA has to stay in the nucleus. Okay, and the reason why it stays in the nucleus is for protection, but also it's a bit too big. It cannot leave the nucleus, and so we've got to get some way for the information from the DNA to get to the ribosomes, right? Because that's where the proteins are made. And where are the ribosomes? Remember, some of them are on the endoplasmic reticulum, some of them are in the cytoplasm, and that's where our RNA molecule is going to come in. So let's kind of talk about the differences between DNA and RNA. RNA is a nucleic acid, so it's still made of nucleotides, and it's the messenger that basically kind of takes the message from DNA to the ribosomes. And the RNA produced during transcription is structurally different from DNA in three ways. Okay? First of all, the sugar in RNA is ribose, and the sugar in DNA is deoxyribose. Number two, RNA is single-stranded, whereas DNA is double-stranded. We can see that here. All right, here's the double-stranded. And the final thing is that RNA contains a base called uracil instead of thymine. Okay, so uracil is going to replace thymine. It's going to still bond with A. It just kind of takes the place of it, and that's one of the three major ways that they are different. So, kind of looking at this, all right. We take a look. These are all the same. This is the sugar. Okay. One of these is deoxyribose, and one of them is ribose. Let's see which one you think is going to be which. Think about the words ribose and deoxyribose. Correct answer. This is deoxyribose. Okay. So if we take a look right here. There's an oxygen right here, okay, in ribose sugar that is not present in deoxyribose. And the reason why that's important is it allows the molecules to form that double strand, okay, and it is essentially, what differentiates the two of them. So RNA molecules have many functions, but in the majority of cells, most RNA molecules are involved in protein synthesis. Okay, protein synthesis is the assembly of amino acids into proteins, just like DNA synthesis would be the assembly of DNA. Anytime we're talking about synthesis, we're talking about putting things together. So what are the different types of RNA? Okay, there's three different types. We've got messenger RNA. Okay, this is the one that you're probably most familiar with. And it's responsible for copying one strand of DNA in the nucleus and carrying that information to the ribosomes. That's why we call it messenger RNA, because it is what contains the message, also shorthand mRNA. All right, and it's just kind of a long strand. Then we've got ribosomal RNA. RNA makes up a large part of the ribosome. There's a little bit of protein in there as well, but it's actually a ribosome is mostly made up of rRNA or ribosomal RNA. And that's kind of got the two pieces to it, a heavy, large unit, and then a small unit. And then finally, we've got transfer RNA, or tRNA. That's what carries the amino acids to the ribosome where they are joined to form proteins. So transfer, it helps to move the amino acids into the correct position. Okay, helps to move it into the correct position. So during protein synthesis, genes copied into RNA are expressed by the production of specific types of proteins. Protein synthesis involves two processes. We've got transcription, and this is where the DNA sequence is copied into a complementary mRNA sequence. What we mean by complementary is that if on the DNA sequence it says A, on the RNA synthesis sequence it's going to say U. And if on the DNA sequence it says T, then on the RNA sequence it's going to say A, so on and so forth. That's what we mean by complementary. They complement each other. They're not the same. They're complementary. They're slightly different, but it's just the reverse. So if it's a G, it's a C, so on and so forth. Then we've got translation. This is the decoding of the mRNA message into the polypeptide chain, or if you remember back, is also known as a protein. So that's where we actually take that code, that piece of mRNA, and change it into the actual protein. So the process of transcription occurs on the DNA in the nucleus, and what happens is that DNA is going to be converted into RNA. A is going to pair with U, and G is going to pair with C. So I'm going to play this video or this demo real quick, kind of talk through it. So what's going to happen, okay, is that we've got RNA polymerase, right? So we've had DNA polymerase, which dealt with how DNA was being formed, right? So we've got RNA polymerase. What that's going to do is it's going to bind, okay? It's going to bind to our piece of DNA. And as it's kind of going through, it's going to read and decode the template strand, right? And it's going to match up G's with C's, A's with T's, C with G, a U, remember, because this is RNA with an A, G with a C, and it's just going to make this piece of mRNA, okay? All right, and it's going to do that. There's some mechanisms that you all don't need to necessarily know that have to do with, you know, stopping and starting this. I'm more interested in you just understanding kind of the basic process of how the mRNA is formed. And the most important part is this right here, okay, where I've got the pairs, right, the complementary pairs forming from the template strand. Okay, so my gene is on the DNA. I'm reading that gene and forming the mRNA. All right, the other process that we've got is translation. And translation is completed by the ribosomes located in the cell cytoplasm. So the mRNA is going to leave the nucleus, and all three parts of the mRNA are going to work together to produce the polypeptide or the protein. And every 3 RNA nucleotides represent a codon, which signify 1 amino acid. So how does this actually work? Okay, the sequence of bases in an mRNA molecule serves as instructions. And we basically decode these instructions by using codons, which are sets of 3 bases that each code for 1 amino acid. And each codon is matched to an anticodon or complementary, that should say complementary, sequence on the tRNA to determine the order of the amino acids. So let's take a look at translation. Let me make that a little bit bigger. All right, so remember we've got our DNA. We go through transcription, that makes the mRNA, and then we go through translation, that makes the protein. All right, so here I've got my ribosome. Okay, my mRNA is going to be joining. You all do not need to know the EP and A sites. I just kind of have that on there. So what's gonna happen? Okay, here's my triple letter code. This is my codon. All right, here's my anticodon with my piece of tRNA coming in. So what's going to happen is that it's going to bind. Here's my amino acid attached to the end. tRNA, the anticodon is complementary to the codon. Right? We've got our next one that comes in. And what's going to be happening is that the amino acids are going to join to one another. Again, these are complementary, so if it's an A here, it would be a U there. If there's a G here, it would be a C. And it's going to move it through the ribosome, right? It's just going to continue to attach them one at a time. All right? Until we finally get finished, and then we end up with our polypeptide with all of our amino acids attached together. And if you want to see the animations on your own, I will have those linked as well. So here's our tRNA, here's our anticodon, and the codon. You can kind of see a little bit better how they match up with one another. And again, it's sets of 3. It's all about sets of 3 when we talk about codons and anticodons. So using a codon chart, it'd be kind of hard to remember if you don't remember. There are 20 different amino acids, and there are lots of combinations of codons. So it'd be kind of hard to remember those. So we have these handy dandy charts that we use. And you will always have access to a chart when you are trying to figure these out. And the mRNA bases or the codons are used to find the amino acids. So we don't use the anticodon, the tRNA. we use the codons found on the messenger RNA when we are using the charts. So let's kind of go through and talk about how to do these. All right. For example, if my DNA sequence is TAC, GCA, TGG, AAT, okay, I'm going to break these up into threes. So my mRNA is then going to be T pairs with A, A to U, CG, GC, CG, AU, TA, so on and so forth. All right. We'll see how these are complementary. Now what I'm going to do is I'm going to look at my three-letter codes. I'm going to look at my codon chart and figure out which amino acids I have. So I've got AUG. So I start over here on the left with the first base. So it's an A. So I know it's here. Right? Then it's a U. So I know it's somewhere in this box. Right? This one and this one gives me this box right here. And then it's the third base. Okay, is going to be a G. So it's AUG. So it's going to be right here, methionine, or MET, which is the shorthand. Okay, and I can just go through the next one, CGU. So CGU, that's going to be arginine, ACC, ACC, threonine, and then the last one, UUA, UUA, leucine. Okay, so here's the next one. I'm not going to say anything for this one, just kind of work through it on your own. And here's the mRNA. And then here is the amino acids. All right. And we will do more practice of this in class. All right, so mutations. All right, sometimes during replication, okay, we talked about this before, an error can be made, all right, and that's going to change the DNA, which is going to subsequently change the mRNA, which is then going to change my protein. So that's kind of how mutations work, all right, is that we've got an error in replication that leads to a change in the DNA that changes the mRNA that's going to change whatever protein is made. So we've got a couple different types. We've got point mutations. Okay, point mutations all basically occur in one or two amino acids, or sorry, nucleotides. And one type of that is a substitution in which one base is changed from one to another. So basically, like from A to G, or so on and so forth. We can also have what we call deletions or insertions. Okay. And these lead to frame shift mutations. Basically what happens is that we lose an A, right? And then all of a sudden, remember we read in sets of 3. So if I lose one of those in that set of 3, now my whole frame has shifted over where I read. My codons have changed, and that's going to be an issue. We can also have some chromosomal mutations, all right? And these can involve deletions, insertions, or versions, but they're of whole sections. So basically, a part of a chromosome is going to be switched over from one place to another, deleted, whatever, and those are obviously going to have major, major issues. And sometimes we can actually have pieces moving from one chromosome to another, and we have those are called chromosomal rearrangements, and those are ways that large-scale evolution can occur. And again, the problem with mutations, or the issue with mutations, not necessarily always a problem, is that they ultimately lead to changes in proteins. Remember, proteins are what do things in your body. So if you change the proteins, you're going to have major changes in your body or in whatever living thing you have. All right, that's all I got for today. Have a great day, and I'll see you next time.